Hello, everyone. Thanks for this great opportunity for us to present our research paper titled Explainable Computer Vision Techniques and Deep Transfer Learning for Tomato Disease Diagnosis. This is Via Drake on behalf of the whole author team from Makerere University Department of Computer Science. Tomatoes are one of the mostly grown vegetables in Uganda due to their high demand both locally and for export. This is why most farmers have invested into the growth of these tomatoes. However, farmers are currently facing a challenge of tomato leaf disease, mainly facing diseases like bacterial spot, tomato virus, and late blight. So this has led to significant losses by farmers. So the early and proper preventive measures need to be taken into consideration, not for these diseases to affect the quality and quantity of tomatoes. So farmers mostly relied on their own visual inspections or advice from agricultural experts and experienced farmers to take these diseases. So by this method can be expensive, time consuming, and not always accurate with risks, because most diseases have the same symptoms. So there's a need for an automated system for early disease detection. Likely enough, recent advances in artificial intelligence and deep learning have offered great potential for early disease detection. So deep learning models have indicated outstanding results across different image classification tasks, plant disease recognitions included. So David P. and Shadara used deep learning models with AlexNet and GoogleNet architectures, which achieved great plant disease classification. Then Smith also employed transfer learning with ResNet 52 architecture, enhancing its capability for tomato disease detection and achieving an impressive accuracy. So as these models are used, the problem is that they require extensive annotated dataset to ensure generalizability and transferability. And such datasets are scarce due to costly annotation processes. And the use of small dataset lead to challenges like inadequate feature extraction, overfitting, and sampling biases. Also, these deep learning models require powerful hardware since they are computationally intensive, hence can be deployed on small gadgets like smartphones and low-cost computers, which most of these farmers have smartphones. Additionally, these networks often operate as black box, providing no explanations for their inferences. So there's a need for a transparent model that farmers can trust. So in this study, we plan to use transfer learning to improve the performance of deep neural networks on small datasets, also integrate explainability into the pre-trained models, and evaluate the different pre-trained models and employed explainability techniques, also demonstrate the practical applications of the proposed approach. So we've implemented and evaluated deep transfer learning models for tomato disease detection, then used different explanation techniques like RLP, GradCam, and SHARP to show the leaf regions responsible for the prediction and compare the effectiveness of different explainable techniques. We've done this through a number of steps, starting with data collection, exploratory data analysis. We've done data pre-processing, splitting the data set, then extracting different features from the data set for model training, then testing the model and evaluating these models we've created using different evaluation metrics like loss, precision, F1 score, and others, then compared the pre-trained models to see which one is best for the task. Then implementing explainable artificial intelligence techniques to solve the black box nature of these models, then did model comparison. So the dataset we used was the plant village dataset containing 54,000 images from 30 different plants, but we focused on the tomato dataset, the tomato leaf images, having 10 classes, 9 of which are affected and 1 that is healthy. So it's having 10,000 images of training data and 1,000 of pre-trained data. These classes include bacterial spot, early blight, and others. So to understand the data set, we did exploratory data analysis. We knew the images, the class distribution, and all those, and two important exploratory data analysis techniques, that is contour detection and edge detection. We did edge detection to visualize and understand the structure of different leaf images, since each disease affects the structure in its own way. So under edge detection, we did an algorithm that we used to find the boundaries or edges of the images, determining where the variation in shape and texture was. Then under contour detection, we have to visualize the outlines of affected areas and veins of leaves images, which are important features that the model used for its prediction. Under the whole process, we did spatial data analysis to provide variable insights into the data set, guiding the pre-processing and modeling decisions for the tomato leaf disease classification task. So for pre-processing, we did data augmentation, that is to increase on the number of images to solve the small data set problems We did this through resizing the images, rotating the images, flipping the images. Then we did imagery scaling to 224 by 224 image size. That is the input size for all the models. Then we created or downloaded the base model, freezed the convolutional base for future extraction, did batch normalization, was done like by freezing the convolutional base, like generally extracting features like edges, texture, color that were used in the model's prediction. So new fully connected layers added to the model are trained on the tomato leaf dataset, allowing the model to learn the specific features that differentiate different diseases. This was done under fine-tuning, allowing the weights of the top layers to be updated while the pre-trained features remained intact. Upper parameter tuning was done using Optuna to get the optimal training parameters, that is the running rate, batch size, and the dropout rate. So a classification head was added for prediction generation. So mode output was interpreted by providing a probability distribution over the possible classes. Like in case can be a probability of 0.1, that is healthy, 0.7 affected, then 0.2 like bacterial. So the 0.7 will be the correct prediction. Then after that, we did the model training. So the models used were EfficientNet B0, ResNet 50, VGG16, VGG19, and Vision Transformers. So these are the results. So we first trained the custom models, that is before transfer learning, and Vision Transformer performed better than all the other CNN models due to its self-attention mechanism to capture global dependencies and relations. So the self-attention mechanism allows the model to understand the relationship between different parts of the image by assigning importance scores and focusing on the most relevant patches. It uses less memory and uses less computational resources. EfficientNet B0 performed next because of its compact and efficient architecture that balances precision and recall. Then we are seeing ResNet performing better than VGG19 and VGG16, attributed to its residual connection, which helps mitigate the vanishing gradient of deep neural networks. Then after transfer learning, Vision Transformer still performed better than all the other models. We used training accuracy, training loss, test accuracy, F1 score precision, and recall as to evaluate these models. Then this is a graphical representation of training and validation metrics for ResNet 50 for the tomato leaf detection, and we are seeing that loss decreases as accuracy increases as the number of epochs increases. So to solve the black box nature of these models, we use 3 different explainability techniques, that is graduated class activation map that uses gradients target features into the final convolutional layers, generating a map highlighting the regions considered in the model's prediction. Then we use sharply additive explanations that assigns each feature of the output a sharp value based on game theory. Then we used layer-wise relevance backward propagation that assigns relevance scores to each input feature. So LRLP propagates the output features backwards through the model network, calculating the relevance from output to input neurons, showing the most relevant features and visualize them into a feature map. So we use GradCam on FishNet, showing the regions that contribute to the model's prediction, then LRLP on vision transformers, and SHARP on ResNet 50. So SHARP shows the regions that most contributed to the model's prediction. So in conclusion, we have demonstrated the effectiveness of transfer learning, tomato leaf detection on a small data set. Use models like Vision Transformer, FishNet, and others, then employed various explainable techniques to uncover hidden insights into the pre-trained model's prediction, thereby facilitating transparent disease detection. And we've seen that vision transformers have performed better than the other models on the dataset we had, uses less memory, then uses less computational resources, and it is lightweight. Hence, can be used on small gadgets that these farmers have, then applied Yaw's reverse backward propagation onto vision transformer to provide explainability. And vision transformer RRP performing better than their counterparts. So we found different limitations, like not all the experimental techniques could be applied on all the models, then the small data set and infrastructure resource limitations. Recommend creation of a tomato leaf data set based on Ugandan tomatoes, then exploring more scalable techniques in age detection and parallel computing. Thank you.